Hi everyone, my name's Greg. I'm an assistant professor in MCDB, and I'm delighted to tell you today about my proposal to develop new therapeutics to treat PI3 kinase mutant cancers. PI3 kinase alpha is one of the most frequently mutated genes in the whole of cancer, but that's particularly true in women's cancers. So in the US, each year there's more than 400,000 new cases of breast, cervical, and endometrial cancers, and 30% of those are driven by mutations in PI3 kinase alpha. If diagnosed early, then current treatments are really effective, but for advanced forms of these cancers, outcomes are really poor. And so there's a major unmet need for new precision cancer therapeutics to treat advanced women's cancers. PI3 kinase alpha is a compelling target because the mutations are recurrent and the cancers depend on them. There are, in fact, 2 PI3 kinase alpha inhibitors currently clinically approved to treat advanced breast cancer, but they face a major problem. They're not mutant selective. So when a patient's dosed with these inhibitors, they will inhibit the mutant protein inside the tumor, but they're also going to inhibit the wild type in the healthy human tissue. And that causes severe toxicity, most concerningly high-grade hyperglycemia. Because of this, clinicians have to dose at sub-efficacious levels, and that brings modest clinical benefit. But there's a huge opportunity here. There are 3 main hotspot mutations that account for the majority of these cases. I've listed them here. There's 2 E to K mutations, glutamate to lysine, and an H to R mutation, histidine to arginine. If we can develop inhibitors that selectively target the mutant forms, but not the wild type, then you'll be able to dose at high levels, not get these toxicities, and thus be able to attack the tumor aggressively. So significant progress has already been made on this front. Several companies have developed inhibitors that are selective for the H2R mutations. These inhibitors are doing really well in clinical trials, so much so that 2 of these have been bought by major pharmaceutical companies for more than $2.5 billion each. But these inhibitors don't work well for the E to K mutations. In fact, no established inhibitors do. And so I'm proposing to develop the first mutant-selective inhibitors that target these E to K mutations in a large unaddressed patient population. So let me tell you a little bit about how these mutations work. In our healthy tissue, wild-type PI3 kinase alpha is normally in an off state. When our cells need to switch it on, phosphopeptides bind to this gray regulatory domain, which switches the protein on. In cancer, these E to K mutations in the lower figure introduce a lysine at that regulatory interface that constitutively breaks that interaction and leaves the protein constantly switched on. And that's what's driving these cancers. So how can we develop a mutant-selective inhibitor when the mutant lysine is a long way away from the active site? So my strategy is to develop allosteric inhibitors that form a covalent bond with the mutant lysine itself. If you can do this, then you'll be able to covalently target the mutant protein in the tumor, but you won't covalently target the wild type because it doesn't have that lysine. And this will give the selectivity that should allow for efficacious dosing. So why do I think this is possible? Well, it's because I've done something similar before. So when I was a postdoc, I developed the first mutant-selective inhibitors of AKT, AKT1. AKT1 is also activated by a glutamate to lysine mutation. And in this case, I also use the same strategy, covalently targeting the mutant lysine. These inhibitors have been very successful, and a biotech company, Terramotor Biosciences, was spun out. We jointly filed IP. We co-published this Nature paper, and they've raised more than $350 million so far and have a derivative inhibitor currently in phase 1 clinical trials. So since then, I've started my own lab at Yale, and we have already established a new allosteric ligand for PI3 kinase alpha. We have solved a high-resolution cryo-EM structure and found that it does indeed bind directly at the site of mutation, non-covalently. So this puts us in a really good position to now convert this non-covalent allosteric ligand into a covalent mutant-selective drug. And so I'm applying for this Blavatnik Accelerator Award so we can really accelerate the synthesis. We need to synthesize lots of covalent analogs to find a molecule, hopefully in 6 months' time, that can covalently modify that lysine to give us this selectivity. And with that data, we should be able to file IP and then seek further funding towards the development of a therapeutic to treat advanced women's cancers. Thank you very much, and happy to take questions.
More of a comment than a question. De-risking covalent inhibitors, as you probably know, there's quite a bit of risk with regards to off-target interactions and hapten formation. What's your plans for de-risking on that?
Yeah, it's a really good question. For cysteine targeting, it's fairly well established, like which electrophiles are safe to use, and there's a lot of examples of those now that work really well. For lysine targeting, we know much less. What I can say for AKT is we actually used reversible covalent chemistry. So it will bind potently, make a covalent bond while the protein is folded, but once the protein gets turned over and digested, the ligand will actually come off, so it doesn't make a neoantigen. So in that sense, reversible covalent chemistry is perhaps a little safer, and so that's certainly a strategy we'll be trying here.
Can you speak a little bit about the competitive landscape, and then I'll pass the mic down because I think there's another question. Speak to the competitive landscape in terms of other startups, not just kind of commercial stage or clinical stage companies, and why you think your approach is unique, and of course, based on your prior experience, how that carries over into this as well. Yeah. So PI3 kinase inhibitors is a highly competitive landscape, and mutant selectivity has emerged in the last 5 years, and now there's a lot of companies developing inhibitors. So in this chart, I'm showing the left two compounds are the the approved inhibitors. They're not mutant selective. These inhibitors I have in the middle, these are all the kind of compounds in the clinic which are mutant selective. Earlier stage, most of them are mimicking these inhibitors. If you look at the patent literature, they are copycats, primarily of Relay and Scorpion compounds, but the Synovation one is actually slightly different. And so they're all claiming pan-mutant selectivity. It's not really true. When you look at their data, they're highly selective. They're pretty selective for the H2R mutation, slightly selective for ETK, but it's not enough to bring a meaningful difference. And so I don't think people have really realized that to the extent that they're pursuing a strategy like ours.
Can you remind me for Terremoto, their AKT1 allosteric inhibitor, was that reversible or non-reversible for the oncology program?
The compounds that we publish in that paper are reversible covalent.
Okay.
The compound they have in the clinic the structure of it's not disclosed, so I can't say too much.
Okay.
But they have 2 AKT inhibitors that they put into the clinic, building off that work.
So I just wonder about overall efficacy if you have a reversible in the setting of oncology.
Sorry?
I just worry about efficacy if you have a reversible in the setting of oncology.
Right, yeah. So in the setting of oncology, you really need high target engagement. Is that what you're getting at? And covalency really helps you there. Yes.
Thank you, Greg.